Okay, well, we'll begin this afternoon's program. This is the chant du cygne, the swan song of the 11th year of this course that's been going on here. And each of those 11 years we've ended different from the usual program. We ended with what we hope will be questions, answers, discussions, disagreements, and whatnot. In other words, more interchange and ideas, because this is an opportunity to ask serious questions that you may wonder about your career, the future, science, where are things going, and so forth and so on. So the logo of this course, as you have seen many, many times, is the most famous bridge in the world, which connects Manhattan and Brooklyn, and was built in the end of the 19th century. Now, the importance of it is that nothing is the same on either side of a bridge once you make it. So the analogy to advances in science on one hand and advances in medicine and public health on another hand is not so far-fetched. And the whole purpose of this course has been to bring together in the same room, the same circumstances, people who deal with both elements of major diseases and health problems. And we are not alone in the sense that there are regularly between 200 and 400 people on the NIH campus who watch live. So if you have questions, either we'll pass you a microphone, or so as not to interrupt discussion, maybe just speak loudly, and our guests will repeat the question so the folks on the outside can hear it. And then it goes through the NIH video archive around the world. And it's really amazing and somewhat gratifying. Some of the sessions we've had have been downloaded. One of them is almost 4,000 times. So the other thing that struck me, just in looking back on this, is over 11 years we've had over 200 of these sessions. And when I look at the people who've spoken and the topics and what I've learned, I hope that you all who have come to these or watched them have shared this enormous excitement at having extremely accomplished clinical investigators at the same time with extremely accomplished basic scientists, and then usually with a live patient who, as one student several years ago put it, puts a human face on a disease. And I, for one, have been fascinated when we've had a little questionnaire at the end of people who've registered in the book at various times, what's good and what's bad. And one of the things that's always at the top of the list is the live patient. And most of the people checking that are PhD graduate students and fellows, which tells us that the idea of dealing with human disease or seeing humans with disease is not something that is relegated to only those who carry a stethoscope or some symbol like that. There is a final exam, which has been on the website now for, I don't know, 10 days or so. It's a multiple choice. If you've been to half of the sessions and you take this and pass it, which is not going to strain your sleeping hours or anything, it's really more informative, you'll get a certificate, which some people find of value to them. It's not a diploma, but it's a certificate. And lastly, those of us who put this together would greatly appreciate suggestions, particularly criticism, helpful suggestions. It's nice for people to say you're doing a good job, but if you have really interesting ideas of people, topics, ideas you'd like to hear explored in this format, it would be of great help to us in putting together a program for next year. Okay, so enough of that. So I'm going to very briefly introduce to you our guests today, and we're really extremely fortunate because each of them is in a position of not only responsibility, but of great leadership in terms of major areas of biomedical research, which many of you are interested in or will hear more about. And we did get some questions from people that wanted to hear answered. But the one common one was, what's really exciting in your field that you can visualize over the next reasonable period of time, not a lifetime, is something that I may be interested in, or that's really exciting. Where is it going? We are all inundated with lots of journals and literature and whatnot and so forth, but in a big picture kind of way, where is all of this moving? Are we standing still? Are we moving? And if so, where and how can I fit into this? So that's the question that we relayed to each of our speakers, but they're going to speak briefly, and then we'll see what your questions are. And please, do not hesitate to ask anything reasonable, because this is an opportunity for it. So our first speaker is going to be Bob Balaban, who is the scientific director of the National Institute of Heart, Lung, and Blood. Now, Bob has a PhD in physics or engineering?
Physiology.
Excuse me. Oh, geez, I should have remembered that. That's where we first met. No, it's not. It's coming back, Bob. Physiology is where it's at. At any rate, in physiology from Duke, and he here at NIH, even before he was scientific director, has really been the leading force in bringing advanced magnetic imaging technologies into the NIH. And it's extended to the point where MRI clinical studies are performed all over the country, but even right across the street in Suburban Hospital in a joint project between NIH and Suburban and other places. And so Bob has developed this, the application of this technology, as well as pursued his basic interest, which is in the bioenergetics of cardiac muscle. So Bob is going to be the first speaker, and then the second speaker is Tom Insel, who is the director of the National Institute of Mental Health. I was just telling Tom that when he spoke in this course a few years ago on autism, that is one of the all-time highs of programs downloaded around the world, pushing like about 4,000. And I would urge you to go to the video archives and listen and watch that. It's an amazing presentation of a complicated subject. So before coming here, Tom was professor of psychiatry at Emory, was the head of the primate center, and his research has involved behavioral type studies linked in with other precise measurements and so forth, and is very much a leader in this field, and who came to NIH as the director, and that institute has, as you can tell from people who've spoken in this course, really does amazing research. And then our third speaker is Story Landis. Story is the director of the National Institute of Neurologic Diseases. She received her PhD at Harvard, where she took further training in neurobiology, and then went to Case Western Reserve, where she is widely recognized for creating an outstanding neuroscience program there, became one of the standards throughout the country, and then came here and has done the same thing. In fact, one of the things she's done that I know a little bit about from hearing of colleagues involved is to take the great diversity of neuroscience research at the NIH, which takes place in, I don't know, maybe every institute, and to bring them together in a bridging exercise so that the sum is greater than the parts, to coin a phrase. And so Story will be the third speaker. And then at about 4:30, John Gallen, who's the director of the Clinical Center, will be with us. And so he'll be the cleanup hitter in this scenario. So John received his MD degree at Cornell, became a leading investigator in infectious disease, came to the NIH at NIAID, is world famous for his research on chronic granulomatous disease, which he basically did all the fundamental work on, and then as director of the largest research hospital in the world. His scope and vision of what's going on, we're just introducing you, is unparalleled. So I think enough of me. Bob, do you want to begin?
So I kind of took the key of what to talk about here, mostly from your questions. And the hardest question I got in there was that with all that's going on right now, would I have gone into biomedical research myself, starting right now? And that's a tough question, but I think the closest way I can answer that is to give you an example. One of my kids. So they share some DNA with me, so there's a link. And my daughter decided, oh, thanks. My daughter decided to go and get a PhD. She accepted at a good program just north of here. And I chatted with her and I said, you know how bad it is out there? As a parent should, in terms of making sure she's making a good decision. And she had all the data and all this stuff I'm going to talk about a little bit as we go forward, and that we all know it's tough out there. She gave me a really interesting answer, which actually probably relates to when I was in graduate school. And the last thing I was really thinking about when I went to graduate school was what my job was going to be. What I was interested in, what I was going to do, right, in graduate school, and what I was going to have the opportunity to do. So her answer was very simple. She goes, Dad, I can go and maybe get a job I like, maybe get a job I don't like, but right now, or I can go in the lab. I can have a great time in the lab. She's a real lab rat, just loves doing experiments, and then come out with a PhD and then maybe get a job I don't want or I do want. And then those 5 to 6 years now, these days, 5 to 6 years, have a great time in the laboratory. And she's been there for a year, and in point of fact, that's what she's found, is that, you know, that's her rationale. And so I guess what I'm saying is I'd probably do the same thing I did 40 years ago, whenever it was, to make the decisions I made. Now, in terms of careers and in terms of what I would go into, I must admit I'd probably right now, knowing what I know about science and where things are going, I'd probably become a developmental biologist. And the reason I'd be interested in developmental biology is that developmental biology is learning how the genome plays out. And the control of differentiation I find fascinating. You all hear about stem cells and iPS cells. Well, we don't know how we're going to control that differentiation because we don't understand developmental biology right now. So I really think, and I'm an integrator, I'm a physiologist, I think really trying to figure out how the peer pressure works in terms of the cell. You know, a cell doesn't wake up one day and say, I shall be a heart cell. It's ultimate in peer pressure. It's all the cells around it that dictate what it's going to become. So that would probably be an area that I would go to, but as we'll see, it may not be where the jobs are as we look at what the future is. I thought it was interesting because most of the other questions have to do with the hot areas or hot topics. Again, my hot topic would probably be developmental biology. But where did our trainees go with PhDs or MDs from the intramural program? And as you can see, much like the national average, we have between 40 and 29% of the people we train are actually going into what we would call conventional academic positions. And in point of fact, this just came out from the AAAS at a wonderful meeting on the future of science training and education and such that just happened down in Washington a while ago. Tenure-track jobs now need to be considered the ad hoc or the alternative academic careers because they are not the norm anymore for new PhDs. And I think that's becoming a realization that there are other aspects one has to look at when going into a conventional PhD position. In point of fact, in our institute, we're starting to ask the question, much like maybe what's going on here a little bit, are we really training people the right way for what the majority of them are going to do? And I know Dr. Tabak was just talking to the BSc members and basically made the same statement that, you know, we really got to look at what our training program is, and are we, since we're only training about 40% to 30% going into the clones of us, that maybe we ought to be looking at that training program. Training for industry, should we really be training people looking for alternatives for funding, that is, starting a business? Or other alternatives, non-government supportive academic positions. You go out and look at a lot of universities right now, and a large fraction of their support is not coming from us anymore. A large fraction is coming from alternative sources. So those are the kinds of things we have to think about. So I talked about my niche, and my niche was developmental biology. But now what are the economists, and what are you actually observing where these alternative jobs are coming from? And I collected a bunch of information from Forbes, Business Week, The Atlantic, and the AAAS meeting was extremely useful. What was surprising, of all jobs you could have, that includes computer sciences, I don't know, business management, whatever, the number two overall growth potential of all jobs is biomedical engineering. And in point of fact, it's even surpassing chemical engineering, energy engineering, all those areas. 62% projected job growth. And really, this is this concept of bringing engineering majors, as we were just talking about, bringing engineering in to support the medical industry. The big areas are imaging, pharmaceutical, and medical devices. Next big area is health information technology, a growth of 20%. Surprisingly, because we've heard all the depression about the drug industry, it really is not really true. But the pharmaceutical industry is still growing. It is expected to double by 2000 to become a $1.3 trillion energy. And this is largely a result of aging in the sedentary population. It's a really interesting theme in the business community. Aging is where it is. Thank God, I need them, right? But it turns out that even on the secondary list, you find geriatric health, again, becoming one of these areas that people are projecting as a real growth area and a driver of both devices, biomedical devices, as well as the drug industry. A huge driver. Another one which came out very high and was a really big topic at the AAAS is data mining. That is, being able to deal with big data. You've heard Dr. Collins announce the Big Data Initiative. It turns out there is a dearth of people out there trained and being able to deal with systems level, systems biology, systems medicine, dealing with these large data sets. And in point of fact, one of the people from Microsoft was there saying, if somebody comes to us with a PhD and understands big data, we hire them with 6 figures, 7 figures, no problem, right? That was a statement from her saying that those jobs aren't out there. The people aren't out there that they can hire. Geriatric health, we talked about, of course, occupational health and safety. So it's interesting when you go through these lists and you say, oh boy, biomedical research is in such terrible positioning. You go through all of these business companies in terms of where they see growth, where they see it. And next to energy and sometimes surpassing energy, biomedical research is there. Why? Healthcare is not going to go away. Quality of life is not going to go away. Wherever the economy is, that's going to be one of the things that drives the market, is people wanting to improve their quality of life. And how you can figure out with your talent set or skill set how to participate in that market, it ain't going away. All the, concerns we have right now, the cold-hearted businessmen are looking at this and going, we're going to put money in this. This is going to continue. This is a field that we can make money at. It's going to improve the quality of healthcare, and this is going to do good for the society. So I don't have a gloom and doom, especially after doing this review and actually seeing what people distant from we are, don't have the passion we have for this science, are saying towards this is something worthwhile doing, this is something that the people are going to support and the market's going to support for a long time. Okay.
So, anyone have any immediate questions? This is one of the more reassuring things.
Well, thanks. Delighted to be here. And actually, following Bob, I didn't bring slides, but we can leave his up. And I didn't do much research on this, but I'll start by saying that in a certain way, this really is in some ways kind of the best of times and the worst of times. And the worst of times piece, having watched this process for the last decade as director, is that it is harder now, to be very clear. It's harder to be a tenured or tenure-track investigator at a university and to get funding than it has been at any time. It's not made any easier by the sequester, but what makes the sequester so difficult is it follows essentially 9 years of either flat or sub-inflationary increases. So we've been losing purchasing power at NIH for a long time. So people who are interested in doing research are finding it hard to get the support for it, at least from NIH. That's the worst of times, and you'll hear a lot about that, even more at universities than you hear here at NIH. There are a lot of people who just over the past weekend, I got 3 emails from senior investigators talking about the possibility they would have to close their lab over the next few months unless they were able to get a grant, and their grants that are into my institute are just outside the gray zone of what we try to fund. And so it's painful. This is a really tough time. And it's impossible not to be an early stage investigator or a student and not look at this and have the rational response that, why would I want to go there? Why would I want to put myself into that position? And that does fly in the face a bit to what you've just seen from Bob and from the sense that many people have that this is a growth industry, there's real opportunity out there. And I think the disconnect has to do with several things. One is that the topography of these careers is changing. He mentioned that 44% number in terms of how many people are going to be in tenure-track positions. If you want to see a really wonderful exploration of those issues and what the workforce is becoming, how it's changing, there's a report that was done for the advisory committee of the director that Francis Collins charged about 2 years ago, and the report was released about 3 or 4 months ago. It's called the Tillman Report. Shirley Tillman, who used to be president of Princeton, was the person who put this together, led the report. It's available on the NIH website, and it's worth looking at because what they did was a really careful review of sort of the pathways. What happens to people after training? How many end up doing postdocs? Of the postdocs, how long does it take? Where do they go after their postdoc? How many end up in industry? How many in academia? Of those in academia, how many in the tenured track kind of position? It's really kind of startling that those of us who are in positions where we're training people, as Bob said, are often not training them for the positions that are out there. And I think that's part of the disconnect, is that there are opportunities. The opportunities are probably going to be quite different 10 years from now than they are today. And they're quite different today already than they were 10 years ago. And we have to be watching how that evolves. My favorite comment about this was from the columnist Tom Friedman, who was giving advice to a graduating class and said, if you're thinking about jobs, everybody wants a job. The best way to find a job is to create a job. And I think that's actually a good way to think about this as a scientist as well. Think about what you're most passionate about. Think about what you could pursue. And then worry later about where that is going to fit into the ecology of science, because that is changing so much. which is my plug to take maybe 3 minutes to answer Wynn's question about what am I most excited about, where do I think the opportunities are from an NIMH perspective. You'll hear a little bit more about this from Dr. Landis, who's going to talk about the NINDS perspective, and there's a lot of similarities here. The world of studying autism, schizophrenia, depression, eating disorders, PTSD, all of those different areas, which is the domain of NIMH, is really changing pretty dramatically. We have a sense that these are really not just mental disorders, but they're brain disorders. And it doesn't happen that often in science, at least in medical science, that the basic science that undergirds a whole clinical area shifts from one area to another. But that's very much what's happening here. We're shifting from 5 decades of sort of psychology as being the fundamental science of understanding mental illness to now it really being neuroscience. And what that means is enormous opportunity for people who are interested in the brain and who want to use the brain to think about how to develop better diagnostics, better therapeutics for this whole realm that we call mental illness. In fact, perhaps we have to change the name of the realm, and maybe calling it behavioral health or mental health is not so useful anymore, since we really want to begin to understand this through the biology. And the biology of these disorders is in fact now tractable. We can do this because we've got fantastic tools, some from the world of genetics, and now we're getting into why I'm saying this is the best of times as well as the worst of times, because the opportunities are there. The ability not just to do kind of rapid and high throughput genetics and to begin to define the subtypes of these different disorders. And just in the last 3 months, almost none of this is published, we're finally getting the architecture, the genetic architecture of these major mental illnesses. And it's so different than what anybody had imagined. There are common variants, there are rare variants, many of them align, so it turns out that when you start to map out where the mutations are, they're also where you see the common alleles. And believe it or not, they turn out to be identifying important genes for neural development. So we're now beginning to think about all these as neurodevelopmental disorders, not just brain disorders. And in a kind of remarkable way, what many of these genetic findings are pointing us to is really a small number of pathways. There are a lot of genes that seem to be implicated in schizophrenia and autism. If you look at each one of them, it may be a little bit confusing. You put them all together and you begin to see pathways, and remarkably, same pathways. So there's lots of overlap between the genetics of autism, the genetics of schizophrenia, the genetics of epilepsy, and the genetics of intellectual disability, all neurodevelopmental disorders. A lot of excitement in terms of cell biology as well. The problem we faced is when trying to understand the biology of neurodevelopmental disorders when the symptoms start at age 18, and the action, the actual place where the cause might be in utero or in the first 2 years of life. How do you get at that? You can't rewind the clock. But you can, taking a page from Bob's interest in developmental biology, you can take cells from an adult and begin to de-differentiate and redifferentiate them in such a way that you can almost begin to create an in vitro model of many of these diseases, which is really kind of remarkable. The genetics is complex. The cell biology may turn out to be a little more tractable. Using induced pluripotent stem cells is going to be an extraordinary opportunity for understanding neurodevelopmental diseases. And then, of course, the big explosion is not just at the molecular or genomic level and the cellular level, but at the systems level, where through the changes in technology and the ability to do neuroimaging, we're really getting these spectacular first-time pictures of both the human brain and what it looks like in terms of the wiring diagram, the connectome, as we call it. The President's new brain initiative promises to bring even better technologies to both look at the brain in a static way, but also to begin to then decode the language of the brain dynamically and to understand how the brain is processing information. We've had already fantastic advances in being able to image the nervous system in a kind of comprehensive way for C. elegans and now for Drosophila. And we're beginning to do this for circuitry in mice. But what we haven't been able to do is to get the same kind of resolution in the human brain. And the hope is that with this new brain initiative, we'll begin to develop just those kinds of technologies. So we talked about genomics, a little bit about cell biology, a little bit about imaging and this ability to, for the first time, understand mental disorders as brain circuit disorders, which is really a spectacular new opportunity for us. The last thing I would say is that as a student or as an early stage investigator, it's not just that the ecology of the job market is changing, but the ecology of science is changing in a really fundamental way. A story and I sat in a few weeks ago on a presentation on the Human Connectome project. And this is a project that we're both funding through the Neuroscience Blueprint. It's about $30 million a year. And it's being done mostly at Washington University and University of Minnesota and Mass General, where they're creating some new tools to do this, to really define the wiring diagram of the human brain in a comprehensive way. But the WashU group is doing 1,200 healthy human subjects, 300 twins embedded in that. And they're putting everything on the web. A lot of cognitive data, ultimately it'll be genomic data, and then these extraordinary brain images with all of the phenotyping embedded on top of them. They've just done the first 68 subjects, and you can go to that website, the Connectome website, and it'll blow you away with how much data one could begin to interrogate. The message I thought as we sat at this presentation was, well, the first thing I thought was, gosh, was I born 30 years or 40 years too late? Because this is really cool. But it is such a gift for students or for postdocs who, for the first time, will not have to spend 4 or 5 years collecting these data. They can begin to mine what's there, and it's going to take an army. It'll really be crowdsourced to be able to even understand everything that's being put up on the web. So there are going to be spectacular opportunities for discovery for people coming up at this point in time without even having to do the experiment, simply being able to mine the kinds of treasure troves that are being put into these databases for imaging. There are also some for genomics, now that all of this is being made public. So it really is, in so many ways, scientifically the best of times. The opportunities have never been better in that sense. And you will hear a lot of gloom and doom, especially from people who are more senior. It sometimes seems to me the further they are in their careers, the more gloom and doom you hear, which may tell you something. I have a friend who said to me recently, you know, science progresses one funeral at a time. That is not a sentiment that I find particularly, in my age, particularly optimistic or hopeful, but it does remind us that the thing about science is it's always being renewed. It is always changing. And for students coming on now, the opportunities to do really fundamental work, discovery science, are absolutely better than they have ever been. And I will let Dr. Landis take it from there.
Do you want to give me the microphone?
Well, yeah, you can have the microphone.
So I think Tom has made a number of really interesting points. I'd like to say a little bit about when I was a graduate student. I was really interested in cerebellar development and thought it would be great to understand more about how neurons survived or didn't. And my tool was a naturally occurring mutant mouse called Nervous. At the time, people in the 20s, people bred large numbers of mice, and every once in a while there'd be a mouse that had some odd behavioral characteristic, usually cerebellar, so you could pick it up. So this was my experimental animal, and I spent 4 years, I finished in 4 years, which was a miracle, actually doing anatomical and histochemical studies on the cerebellum of this mouse, which lost Purkinje cells. And the only thing I found, which Bob will find interesting, is that instead of elongated mitochondria, they had these big balloon-like mitochondria. Now, if I were interested in survival of Purkinje cells now, I could, first of all, if I took that mouse, I could get the gene that was responsible in no time at all. In fact, it has been cloned, and I don't remember what it was. It was something completely unexpected. But I could also design a mouse. I could design a mouse that would lose one of the neurotrophic factors at a certain stage in development in a certain population of cells. I could do elegant experiments that would allow me to dissect the pathways that were responsible. So the tools are amazing. And Tom mentioned induced pluripotent stem cells. I was the person who ran the stem cell task force at the time when human embryonic stem cells were so controversial. And it turned out that with human embryonic stem cells, which came after mouse embryonic stem cells, you could actually get a Erlenmeyer full of motor neurons, human motor neurons, or of dopamine neurons, of retinal neurons. Extraordinary opportunity, and to take advantage of all we knew about the normal developmental signals in the brain and then translate them into a sequence of factor treatments, and then to have all the human motor neurons or all the human dopamine neurons that you could ever want to study in a dish. Never imagined that that would be true, and then to be able to take skin cells and turn them into pluripotent stem cells like human embryonic stem cells and take them from people who have mutations and learn why dopamine neurons die in people who have inherited forms of... Amazing, amazing. And then to use those to screen for therapeutics that might be of interest. And a wonderful paper that came out in Cell Stem Cell looking at ALS and documented the discovery of a new family of support factors and then replicating 2 failed experiments with ALS patients that the mouse models had suggested that these 2 compounds would support and were going into human trials. But if you did it on the human ALS motor neurons, you found out that those 2 compounds weren't successful at all. So it's an amazing time. Everybody, I think, worries now, is there a seat at the table for me? And I think your daughter's decision, I'm not going to worry about the table yet. I'm going to have a good time at the lab bench, and if I'm successful, things will pan out. I can tell you, it never occurred to me to worry about what I do after I got my PhD. I just had to worry about getting into graduate school because at that time, the admissions committee at Harvard, one of the members asked me, well, if we accept you, will you actually finish your degree? Because everybody knows women get married and then they have families and they drop out. So that's another reason why it's a really good time, because there's not that same set of issues. So my postdoctoral fellowship was impounded by Richard Nixon, tells you how old I am. Wasn't personal. Every single fellowship was impounded that year. But then the next year we all got those fellowships. And when I sat on study section in the early 90s, 1 out of 10 grants got funded. So you'd go with your pile of 12 grants and you'd pick out the one that you were going to go to the mat for to get funded. So it's been worse before, and it's always gotten better. I don't often say that as much now because I keep thinking it should have gotten better. But given the opportunities for research and the need, I'm convinced that this is, it is going to get better. Now, I also want to put in a plug for basic science. Tom made a very compelling argument for why there are extraordinary opportunities in developing better understanding of psychiatric diseases, and the same is true of neurological disorders. We now know that there are genes for neurological disorders like Parkinson's disease we never thought was genetic. We've learned that there are a number of genes which cause amyotrophic lateral sclerosis or Lou Gehrig's disease, and that allows us to fractionate patients according to the genetic basis of their disease, and those genes will give us clues about sporadic neurodegenerative diseases. But the basic science underpinnings, and I can give you one example. So one of the genes that causes Parkinson's influences the activity of the proteasome. Nobody would have guessed that. Really interesting. So what do we know about the proteasome? Well, we know a huge amount about the proteasome because of basic science research funded by NIGMS for a decade or longer, which then the people interested in disease can say, ah, proteasome, read those papers, and put together a reasonable hypothesis. So green fluorescent protein. People use green fluorescent protein now to label neurons so they can record from them. We can do work in neural circuits using channel rhodopsin optogenetics, because somebody was interested in these weird proteins that were light-sensitive channels. And using those tools, we can look at the role of different populations of neurons in circuits. We can inhibit them or excite them and see what role they play. And all of those tools have required basic science findings, curiosity-driven, not worrying about where it's going to go, just asking and answering a really interesting question. And so I know many people now believe, and we see this from data we've been collecting, many young people who are starting out their careers think, if I'm not working on a disease, I'm never going to get funded. Well, it's not true. It turns out in my institute, with grants reviewed by Center for Scientific Review, you have a better chance of getting your grant funded if you're doing pure basic science than if you're doing some cobbled together translational project. And I think one of the reasons for that is the people who are doing pure basic science are passionate enough about that question and about that science to say, I'm not going to follow the crowd, the translational crowd. I'm going to pick this question, which I think is really important and really exciting, and I'm going to pursue it with all the tools I have at my disposal. So I would say for any of you who are in training and are worried, oh my God, I got to be studying a disease, or I've got to be studying traumatic brain injury, so I'll have a rat model which we've hit over the head in order to mimic. If you're passionate about that, that's great. But if you're not passionate about it, don't feel that that's what you have to do. So Tom didn't talk much about investments in training. For NINDS, we are funding just as many trainees as we have in the past. These are people who are on training grants or who have independent fellowships. And that's because we believe that people should have the opportunity to pursue their dream. And we are, when people are moving from postdoc to faculty, fellow to faculty, there are award mechanisms, the K99, R00, we fund a bunch of those, and every one of those that we funded, except for 2, have gotten jobs, really good jobs, and the 2 who didn't had geographic constraints which didn't permit them, they got reasonable jobs, not as good jobs. And then when you're applying for your first R01, there's this notion that we should give those people a chance as well. But you don't have to be a clone of Bob or Tom or me. There are lots, and there are 3 of you sitting right there whom I know really well, who have chosen to use your PhD in a different way. And the one thing you learn with doctoral training, once you get past the coursework, is you learn how to ask a question, how to develop tools to answer the question and then analyze the data, and when they don't come out the way you expected, to pick yourself up and ask it in a different way. So it is extraordinarily good training for a whole variety of tasks that you might take on. So I agree with Tom. I wouldn't say it's the worst of times. It's been bad before, but it's certainly not a great time. It's better for people who are training and starting out than for people who may be close to finishing. But that the tools that are available to allow us to answer questions that we never even dreamed we could answer are remarkable, and I think to not gamble and pursue this would be too bad. But you have to be passionate. If you're not passionate, forget it. John. John will make the argument for translation. And you won't get the last word, I don't think, because we all get to sit up here.
Thanks, Story. So I represent a slightly different background than some of the other speakers, but I think the three speakers that have preceded me have really done a fabulous job synthesizing the issue and talking about some of the things that should drive all of you. And I'd like to start my comments telling you a story about when I was probably in about 5th grade. At that point in my life, I was really, really interested in the clarinet. And that's what I did much of my time. And a guy who was a neighbor of ours and was a close family friend was a guy named William Schumann. Now, William Schumann was a composer, and he won the Pulitzer Prize for something called the New England Triptych, and he was also president of Juilliard and later became the first president of Lincoln Center. And he used to walk in front of my house every night composing, and one night I was out there and he had heard me playing the clarinet. He says, "So what do you want to do in your life?" I said, "Gee, I think I'd like to play the clarinet." And he looked at me and he stopped and became very serious, and he says, "Let me tell you something. Unless you can't live without it, make it your hobby." Now, he was telling me something, which he was, but I think that you could say the same thing about science. It's something that you should have the passion that Story talked about, and it's something that you just do it because you love it and you pursue it. So what do you pursue? I can remember when I came here in the early 1970s, so you know how long I've been around, and I was introduced to Julius Axelrod, who had this little lab, not so little, but he had a little metal desk in the middle of his lab, which was a prison desk made by the prisoners, nothing fancy. This guy just won the Nobel Prize. And somehow my mentor, who was a guy named Shelley Wolf, had brought me down there and to talk to him about some stuff. And during the discussion, he said, "Let me tell you something. It takes just as much work to do an unimportant project as it does to do an important project. So think carefully about what you're going to pursue." Maybe he was giving me another message, but he said that, and it had an important impact on me, and it's something you should remember. So I would say have passion, and as you design your studies and as you think about what you want to do, pursue something that you think is really important, and give a lot of thought before you start doing something. And it doesn't matter why it's important. It may be important because it's the most fundamental basic science in the world, and it also may be important because you think there's an urgent public health need or clinical need that you're going to address. So I got a medical degree. I don't have a PhD. The closest I've gotten to a PhD is I married a woman who had a PhD, and we had some opportunities during our life to do some studies together. When I was a student, they kept saying to me, "Get as basic as you can get in your research." This is for clinician. Understand the language, speak the language, and do as much as you can and surround yourself with people who hopefully are better than you in the basic sciences, and then you're going to do some good things. And so that's what we tried to do in the labs that I worked with, was to build teams. And we haven't talked about team science tonight, but that's obviously certainly in translational research critical. Teams of people who are PhD basic scientists who can add a dimension and a quality to the work that's being done. And so one of the questions you asked was, you know, where do you think your science should go, or what would you like to do in your career if you were thinking forward? And I have really two areas. My own lab area, which is I was trained in infectious diseases, and I have a passion for host defense, and I got interested in phagocytes as important. And then I have this job as the director of the clinical center, which is a different kind of a challenge. And let me tell you a little bit about each of those. So when I was a young fellow, I worked on neutrophils primarily, and a lot of people turn to me and says, what are you studying those cells for? There's lots of them, and they're sort of boring, and they go and they do things, and they're short-lived. And my reaction was, well, aside from the fact that I think they're kind of fun to look at and they move around, the most common cell in the white cell in the body, and when you lose them, you die. I mean, they must be important. And so we've spent a lot of time studying them, and a lot of time studying rare diseases, which are important models. And one of them that I've worked with our team of some PhD scientists who helped unravel the genomic basis of the diseases and the structural biology is chronic granulomatous disease of childhood. And most of the work for 20 or 30 years was trying to understand why these children got infections, which they do, and how to boost their immune system. And more recently, what we've been working on is an observation that what do these children not get? They don't get atherosclerosis. So the defect in this disease is that they don't make reactive oxygen species, they don't make superoxide and hydroxyl radical and hydrogen peroxide. And yet, if they survive their infections and they live into their 30s and 40s and 50s, which many of them are now doing, we've recognized that they don't get plaque formation in their carotid artery, things that we can do today, which we could never imagine doing 20 years ago because of the great imaging technologies and the application of the things that Bob Balaban started talking about. I personally think understanding how inflammation relates to so many chronic diseases is a great opportunity. How does it relate to Alzheimer's disease? You know, some of the cytokines like interleukin-1 critical in Alzheimer's disease or not? Those are good questions, in my opinion, to ask. And you could go on and on for most diseases like arthritis and heart failure, et cetera, as people are doing. So that's one opportunity that's a little different from some of the ones you've heard about. Now let me tell you a little bit about clinical research and translational research, which almost got a dirty word tonight, but I think it's, you know, health is what this place is. It's the National Institutes of Health, and you have to keep that in the back of your mind, I think, no matter what you do. And translational research is part of that spectrum. And that's what people care about. People care not only about what new medicine is going to come down the road next week to help my disease or my family's disease, which we all worry about, but why is it so expensive? You could say, should the NIH and its great minds be thinking a little bit about some of the social problems? And I think the answer is yes. Whether you want to do that or not, that's your business, but somebody's got to do that, and somebody ought to do that today as we move forward. So I think I'll end by saying that one of the things I've observed in talking to and looking around at people is having flexibility in what you choose as your career paths, and realizing that you don't have to stay doing the same thing you're doing today 30 years or 40 years from now, but you may, maybe you'll do something different. That's fine. That's good, and it's healthy. So I would keep an open mind as you go down the road. And I guess I'll end by saying I agree with the comments of Story that it may be a tough time today, but it's cyclical, and it does change. And I can remember vividly when Harold Varmus became the director of NIH, he looked at us and he said, we got to do more with less. I mean, that was all we heard about for months. And then there was more, and we stopped thinking that way. And, the NIH budget doubled, and things were great. I don't know that we're going to double our budget in the next 10 years, but I do feel that the country and the world are passionate about what you guys do and what you want to do, and if you have the passion for it, you should do it. And you'll figure out a way to have a good life, just like Bob, I guess, is encouraging his daughter to do. And I can tell you that I have some 8-year-old and 14-year-old grandchildren, and they ask me, what is this business of science? Should I go into it? And I say, if you have the passion, do it. So, thank you.
Good. It's a little bit like yours. I'm a physician scientist, a little different vintage, but my story is of a PhD student named Peter who was studying cardiac muscle myosins. And this is when I was chairman of physiology at Tufts. And I don't know, just one day I said to him, Peter, have you ever seen a human heart? And he looked at me and said, no. I said, would you like to? He said, yeah, I really would. So I called up a friend who was a cardiac surgeon across the street who was doing coronary bypasses many times a week. And by that time, it had lost its drama. So he was operating without an audience most of the time. So to have a bright young student come over. So Peter came back quite a few hours later, and he said to me, what is it when the heart goes like this? And I said, well, that's called fibrillation. He said, what's that? I said, look, here's a book. I want you to go read about it, and then come back, we'll talk about it. So he came back several days later. He was very enthusiastic about the whole thing, and he described to me circle movements and the parasympathetic and the sympathetic and how things were working out to influence the formation of fibrillation. And then he said, at that time, I forgot how many cardiac muscle myosins were known, but there was a bundle of them, and it wasn't clear what they were really doing. And Peter said, maybe some of these myosins are under neural control. Now, that was a bright light. At that time, I had friends working at the forefront of cardiac muscle myosin, and I never heard them talk anything about neural control. Well, Peter got all excited about it, did some research, published a wonderful paper in Nature about it, and wound up in the cardiology unit at the Mass General Hospital. And today, he's an associate professor at, he's more than that now, at Harvard. And he's not an MD. He's having the time of his life because many of the very bright young cardiologists who've come into the field are trained more in the molecular area, and the funeral hasn't taken place yet, so the folks they're working with may not be quite in that tune. So he's had a wonderful career and opportunity. And he doesn't want to be a cardiologist, but he certainly is interested in what the problems are that confront the cardiologist and the patient. And it's this area of sort of bridging from back and forth that I think gets a little short-sighted when people talk exclusively about basic versus translational. It's really a communication business, and the whole idea of communication across these two technically very different approaches to it. The communication area is something that's been neglected over the past 20 or 30 years. There's an assumption that if there's money out there in a given area, people are going to flock and do it. Well, to some extent that's true, but it's also true that if you're able to understand something about what people interested in disease are talking about, you come up with your own ideas, which may turn out to be very basic after all, or not. It doesn't matter. But the communication aspect of it. And in a way, that's what we're sort of been trying to do here. So, please, you have questions you wish to ask our panelists? Yes, please speak up loudly. Huh? Go ahead. Turn it on.
That's a barrier. That's not a, so I think that is one of the limitations uniquely for researchers at NIH, is that in anybody who has an entrepreneurial bend, I actually divert them away from here because that is difficult to do. I'm arguing that if you go out and look at universities now, many people have private concerns associated with even their NIH grant funding, and you've all probably saw the 60 Minutes where you walk through a guy's lab and he says, now I'm in my company and now I'm in my R01 funded laboratory as he walks through his laboratory. But I do think that is a direction you're seeing outside doing as government funding is reduced. I think to Story's point that I think the government's got to be the major funding of basic research because the payback is too distant and too difficult for venture capitalists to get involved with. So I think that's a major role of government, and one of the most important things that we do is the basic research. But as you get to where it can be a little more applied, both from an engineering or medical, then I think those tacks to take this translational piece and move it into the private sector for development is something we may have to take more seriously. Certain our colleagues on the outside are taking that more seriously and taking that route more seriously.
Use the microphone.
I'm sorry. So let's say that somebody does this. If there's some sort of arrangement where the government could take a cut from whatever business they start, then wouldn't that be...
So the government's not in the business to make money.
Okay.
Right? But, you know, the biggest, I mean, our biggest reason for patenting anything and even using IP is to make sure that your business partners are protected in their investment in terms of developing technology. And, you know, we have SBIR mechanisms to support small business, and I think that's probably been underutilized or not well utilized in the past, and it'll probably be utilized better as things get tighter and investigators look at that role and maybe using that to support aspects. But it's difficult when you're using the public dollar to support an individual's efforts when they're being funded, their salaries directly coming from the government.
All right.
Do you have any other? Yes, please speak up.
In the first presentation, several individuals went into academia. All of you have mentioned how there's such a flat line right now with government funding, but in academia, a lot of times tenure and promotion is tied to funding, and certainly a lot of times NIH funding. Would you share your thoughts, even though you've been in academic positions before, would you share your thoughts on whether you see academia needing to change in terms of some of its guidelines and criteria for tenure and promotion, given that some of the funding from NIH is tied to that promotion and tenure, but it may not be there?
Well, I think each of us can, I think all of us have been in academic positions at one point or another. Maybe not Bob, but certainly Story and I have done this. And this is really changing. The meaning of tenure is very different now than it was 20 years ago. In many places, particularly some of the less academic sites which often are more research intensive, Cold Spring Harbor, Salk, Scripps, Genelia Farm, they don't have tenure. That's not seen as something that's an advantage either to the institution or even perhaps to the people who are coming to work there. For universities, tenure was really originally developed as a way of protecting academic freedom, not as a source of a sinecure or of some sustainable salary resource. What is happening is that the challenge is greater than ever, and it's not so much in just getting, for instance, a K award, as Story mentioned, or an R01 grant. For many places, you have to have 2 R01 grants to get tenured, and that is particularly difficult. So people really are struggling, and what we're seeing are people who we support making it to the first stage of an academic position, but then really having a hard time at that 5 to 10 year period when they come up for what is a tenure decision still in some academic places. So I think you're right. This is tough. Are universities changing what they're doing? Some are. They're at least talking about it a lot. And one of the places where you see some universities going is A, developing other kinds of academic lines that don't involve tenure. B, thinking about whether the criteria for promotion need to change. It's really difficult, for instance, for people who do team science to get recognized for what they've contributed, and that's a challenge in a world in which NIH increasingly is looking at interdisciplinary or multidisciplinary or team efforts in science. And then C, I think the other place that's probably the most different is trying to figure out whether the whole process by which promotion takes place can involve other factors beyond simply the amount of money that you bring in, and can look at other kinds of contributions. But we should be clear, to go back to Bob Balaban's original slide, that whole process of someone finishing a postdoc, getting an academic position, getting on a tenure track, becoming tenured, developing a lab, spawning the same people to do the same thing, that's not what it used to be. And we're talking about less than half of people getting PhDs in biomedical science going that direction. So it will have to be probably less and less as the pipeline and the pay line readjust. We're going to see the need to change a lot of those expectations. Story?
A real brief comment, though. We've all now trained people at this table who have gone into the alternative careers. I have to say, I would say a vast majority of them are ecstatic in what they're doing. They're doing science, but they aren't necessarily fighting through the R01 system. They may be arguing with a boss or arguing with a corporate or arguing through different types of problems. But they're all ecstatic about doing science. They're all ecstatic about the career decision they make, though some of them come back sheepishly because I thought they could make it, they could clone me, right? But they chose not to. And so I think that it's not just that it's a bad thing, it's just a different thing. And it is very satisfactory, and I think they feel very fulfilled in what they're doing, and they enjoy going to work every day.
So one of my postdocs went to teach at a small liberal arts college, has R15 funding, which is meant to allow a person to have research and to engage undergraduates in that research. Got promoted and tenured relatively quickly, and as Bob said, is actually extraordinarily happy. Graduate student of mine did a postdoc, quite quickly moved into administration and is dean of something at Mass General, and actually happier than she was when she was working for me in the lab. Has nothing to do with me, I'm sure.
You can tell us.
You know, and has a couple of kids and is very fulfilled. And a lot of the kinds of problem solving that she does in that role is informed by the kind of, you know, what's the question, what are the possibilities for answering that question, how do I solve this problem? So lots of different ways. And I would say, Tom, your glass may be half empty and only half of them are doing research. I would say my glass is half full, and half of them are doing research.
Yeah, so I have two of my best graduate students ended up going to law school because they were interested in technology transfer and IP issues, and both have done extraordinarily well enough that not only would they not need an R15 or any other NIH support, they are now able to support science philanthropically. So there are lots of careers that you can go into. My father used to say that going to medical school was a part of a basic education, but increasingly, you know, you could even think of getting a PhD as sort of a basic process to learn how to solve problems. I really love Story's comments about it's a chance to figure out how to actually solve problems. And it's training in that you don't get in many other areas. It's quite different than medical school.
And to deal with experimental failures. So if it doesn't work, you don't quit. You just figure out you have to change the calcium concentration or whatever.
That's
just,
what it's. That's
just to
that's
build
what
on that
training's
for a
about.
second. I get really annoyed when people say, "Oh, we're training too many PhDs," because I really feel that what you guys were just saying is important, that we have these types of educated, trained people in problem solving and what the PhD process is supposed to be out there in the community. And maybe we'll get more sensible voting practices as well.
John?
Yeah, well, one thing I would add for you, and I see a lot of young people, which is great, as you go out there and you start doing things and you probably become successful, there's a price you pay for your success, and that is then you're asked to do all sorts of things that at one level might be viewed as wonderful, like becoming a dean or you know, going and starting or being involved in a company administratively. My advice is that when those opportunities come, think carefully again about what your passion is and what you want to do with your life. Because I've also seen people who've become deans and for a moment thought that that was the best thing they could possibly do. But after a few weeks of signing their name, you know, over that signature that says dean of something or other, they suddenly realize, gee, that's not really what I want to be doing with my life. And then they have a problem. So you need to make your decisions carefully.
So let me ask a question. For some of us, at least, when we got involved...
Could you use the microphone?
Well, for some of us, when we got involved in biomedical research, two things were very different. One is that even within the clinical department, there was incredibly close linkage with particularly biochemistry, which in those days was the... But, I mean, it was not just occasional conversations. People actually, like, went to rounds and all kinds of interactions. You felt part of a community that you would discuss problems and even ask questions that may have sounded silly but led to some enlightenment. So one question is, within the current scheme of medical education in the medical schools, I get the feeling that that kind of unified environment is rapidly disappearing and has been for some time because of the demands to teach specific topics and that huge mass of information that supposedly every physician is supposed to know. And the other question that I wanted to ask that you might consider is, in those days, medicine and science was not global. At least it wasn't as global as it is today. So when you read, like in last week's Science, 4 different countries are doubling their budgets for research and development. Now, granted, they're much smaller, but nevertheless, what they're doing has their eye on the ball. I agree with the wave theory of ups and downs, having lived through many of it in my career. But I'm a little concerned that now in a global environment where support of science is increasing in many parts of the world and stable or not changing much here, is this something one should be concerned about, or just focus on what you want to do and your passion?
So let me take a crack first. And in terms of what your question is really, what is the medical school environment like? And it depends what school you go to. So if you go to a school like Stanford, I've been phenomenally impressed at a program they have merging their engineering school with their medical school. So what they do is they take students who are early in their career, one engineering student and one medical student, and they make them go into the hospital and walk around and talk to people together and figure out a problem that needs to be addressed, and their assignment is to address it. And Harry Greenberg, who's the dean of research out there, recently described, in just 3 or 4 years, they have 2 or 3 major inventions that have come out of that partnership, and that comes from looking at clinical situations, talking to the care providers, and understanding the problems, and then translating it into the bench, and then going back to the bedside. And so I hope that continues, because that's really what makes clinical research exciting, and it depends on the people. In terms of global stuff, I've had the good fortune of being involved over the last 5 or 6 years with a number of things that are going on in clinical research training in China. And we brought our training programs that we teach here about 8 times to China over the last 5 years. And it's been remarkable to see the transitions that occur in that country, all the way at the very top. I mean, I had the good fortune of interacting on several occasions with the Minister of Health there, who's a scientist. And I can still remember in China the day they passed their healthcare reform, which was very close to Obamacare. I was with him, and I said, congratulations on your, this has been a great week for you, for your healthcare reform. He says, yeah, it has been a great week, but not because of that. It's been a great week because my wife and I just had a paper accepted to science. I think the opportunity for partnerships globally is very exciting and very real, and to get engaged in medicine in the developing countries in particular is phenomenally exciting, and is something that more and more young people are asking and begging to have that opportunity, and more medical schools are making those opportunities possible. So I think it's great, and I'm all for it.
I want to emphasize what John was saying earlier. I don't think you're right that there's this separation in all fields. Maybe the biochemists aren't as close to the clinicians, but certainly the engineers, as you pointed out, are. And the tremendous growth we've seen in biomedical engineering, the work that we're talking about in terms of imaging, the Connectome, this is an engineering problem, okay, to get that data. Now the interpretation will be up to the neuroscientists, but to get the data is really a huge, massive engineering problem. There's wonderful teams developing all over the country, the world, to do this. So I think the explosion in biomedical engineering that we're really witnessing occurring right now is that marriage, and a very successful marriage, the Stanford being another model of that. So there's at least one area where that communication, I think, is very, very close. And I think we heard the stem cell story. As all of a sudden, I've told all my cell biologists, why would you ever study HeLa cells again, right? Why don't you go and talk to some of the clinical people, find some cells from patients who have interesting diseases that you're studying in terms of your basic science studies, and why not use one of them at least in your models? And that's happening at a remarkably quick pace, where people suddenly realize, now I can do the same experiments, transfections, everything I used to do in this cell line that didn't really nobody cared about, where now I can actually do it in clinically relevant material. So it's I'm a little bit more feeling that there's actually reasonably good communication, and these new tools, both from an engineering perspective and cell biology perspective, are actually starting to bring people closer together, not further apart.
So one of the most exciting projects in the Common Fund, which is a fund of money to support projects that are big and not within any one institute's purview, is a collaboration with DARPA. And the collaboration is that DARPA's making platforms for organs on a chip. Little hearts hooked up to lungs, brains, kidneys, livers. So they're doing the engineering with the platform, and NIH is funding investigators to develop functionally appropriate cells to comprise those organs. So a perfect example of that kind of engineering biology collaboration, and the kinds of things that you'll be able to do with those organs on a chip is do toxicity. Why do liver toxicity in rats or mice or dogs when you could with human cells in the liver on a dish, or cardiac, and also to do some initial drug screening. So instead of doing it on dissociated cells in a 195 well plate, do it in one of these little organoids. So very, very interesting.
I do think, oh, I'm sorry. Go ahead. I was just going to say that chemists are needed desperately, and so I don't know, I still think biochemists are also needed, but we need chemists in drug discovery, and one of the great exciting moments for me is I told you about this kids with chronic granulomatous disease who have a broken NADPH oxidase, which we think maybe protect them from atherosclerosis. I got to go visit Chris Austin's team and meet with a bunch of his chemists, and I said, look, here's what we know about this enzyme. Can you think of building an inhibitor that we might give therapeutically, actually, or prophylactically, to make normal people a little bit like our patients with CGD, with the idea of, gee, maybe that would add to our protection against atherosclerosis, since these kids seem to be protected. And so, I mean, what a wonderful opportunity to sit and talk to these people for an afternoon. And, you know, then they came back a few weeks later and they say, here's a bunch of ideas. Let's screen them. And, you know, that's the way it sort of gets going. So I couldn't design a new molecule to attack the enzyme, but they can think about it. And so chemists are, I think, crucial for the future drug discovery. You probably all know that, but it's...
There are two subjects that I'm very passionate about, and they are data mining and crowdsourcing. One or two of the speakers mentioned these topics briefly. I wonder if any of the panelists would like to talk about the experience with these subjects in their institutes, and what might be the future of them in your institutes.
I think Tom gave the best example. Very soon, you'll have a complete image and genome and neurological workup of people's brains. What are you going to do? How are you going to deal with that, right? It's just the similar type of problem we've run into the genome. It's turned out to be so complicated. So a lot of our projections of how powerful the genome was going to be, yeah, Lord, it's been powerful, but not as powerful as we thought, because it is so complex. There is so much more there in terms of the complexity of control, how the genes are regulated, et cetera, that we didn't imagine when we started the program. So this deal of how to deal with systems biology, as we call it, or physiology when I was a kid, or the dealing with these large data sets to now get some reasonable, testable hypotheses and understanding from those massive amounts of data is really probably one of the major challenges, I think, I tell my students that they're going to be facing. And it's going to take a different way of thinking about, from the reductionist view, where we were all taught, get it down to a single protein. Well, we probably can't think that way for much longer. There's going to be reductionist elements. They're kind of the tools and the wrenches, but how the system works in harmony is going to be is a major challenge, I think, of the next generations in terms of sorting it out. We're going to have all the individual molecular machines pretty well understood here pretty soon, but how they're worked in harmony, how the orchestra works with the clarinet and a tuba, is really one of the major challenges, and I think going forward. So this idea, someone said physiology is coming back. Well, it's coming back with a different name. It's coming back with the name of systems biology, systems medicine, that is trying to put the whole back together as decades and decades of reductionist biology have given us all the parts. We're now trying to put it back together, and it's even more complex than I think anyone feared.
Just a word on crowdsourcing. I think it is part of a very new culture for how science will get done. There are already some rather remarkable stories that are being told. There's a website called Eterna, which is actually spelled out to have part of it be RNA, looking at RNA microstructure, and that has been used by tens of thousands of people now, essentially playing a video game in which the whole business of RNA's three-dimensional structure is turned into an animation challenge. And people who are playing this game are not scientists. A lot of them are actually information scientists, but they're not people who know anything about biomedical research or actually anything about RNA or transcription. They are just trying to play what's a really fun game. And in the process, they've been able to generate structures that actually are pretty close to what nature does. But we didn't know that. We didn't have that information before. And so you see people like Phil Sharp at MIT, who are actually, great RNA biologist, who's actually taking information from these thousands and thousands of people, none of whom, by the way, have PhDs, and who are trying to then give him information that he can begin to use. Some of them are actually presenting it at scientific meetings even, that are taking the whole field forward in a way that none of us could have imagined. So the culture is changing, and where answers will come from, where information will be generated, how that information gets generated, could be very different over the next few years.
Group sourcing data is as complex as a complete image of the brain. In other words, you have all that data, all that input from thousands of people. How do you sort the chaff from the novel idea? That's a whole nother process. And just as complex as biological data are all the ideas flowing in from all those brains out there on the web.
Are there other questions?
I think I would like to ask you a question. A lot of our funding comes from public opinion, and right, knock on wood. And we are all, as you all pointed out, quite concerned about the funding level of the NIH and so on. So how, perhaps you can tell us how each one of your institutes address the issue of making this public opinion, especially about the NIH and the contribution of NIH to science and funding of research, available, and how do we gather this momentum now that the funding is low to make NIH more visible and make NIH more important in the public eye for funding?
Well, I think each of us can say something about that. As an institute director, part of my job is talking to the public. It's not just that much of our funding comes from the public. For NIH Institute, all of it does. All of it's appropriated. All of it is taxpayer supported, and we work for them. We are, and our job is to be good stewards of their investment in us. So I'm responsible for about $1.5 billion a year of taxpayer support. So I take that responsibility very seriously, and I think part of my job is to explain how I've used the money to produce information that's of value to the public. So I do a lot of communication on everything from giving TED Talks, which are actually so far the best way I've found of getting a story out there, to having a blog, which gets picked up in ways that you could never imagine, and sometimes completely misinterpreted, as today. But there's a lot of what we do that has to do with trying to explain the value of science, both basic science, clinical science, and helping the public to understand why it is that this is a a good investment. And by the way, one of the most important messages to convey is that the money that goes to NIH is not a cost, so it's quite different than the money that goes to CMS or that goes to HRSA or that goes to many other parts of HHS, which are used for entitlement programs to support healthcare. Really important. Those are costs. The money that goes to the NIH is an investment to try to reduce those kinds of costs. And part of our responsibility is to explain to people why that's such a good investment and why, as healthcare becomes even more expensive, it's even more important that we make investments in making sure we know how to do this efficiently and in a cost-effective way.
So I think one of the misperceptions is that devices come from device manufacturers and pharmaceuticals come from pharma companies. But if you look at virtually any device or any drug and you track back its origin, the most important discoveries were made often, almost always, with NIH dollars. And I've often thought that if I could get every stroke neurologist who treats a patient who's had a stroke with TPA to say the reason you're leaving the hospital with, as a third of the patients do, with no sequelae from your stroke, if they have a stroke center, and if you get there in 3.5 hours, is because NINDS ran a trial in 1995 that showed that this was of benefit. And stroke neurologists aren't going to do that, and medical centers aren't going to do that. But if we had more effective ways to say deep brain stimulation for Parkinson's disease makes a huge difference for patients in the mid-stage, that came out of decades of research on the circuitry, the use of models, a primate model for Parkinson's, which came out of research here at the NIH done by Irv Koppen on MPTP, and work that it was in the neural prosthetics program funded by NINDS, which got put together by Medtronics, and actually a kind of crazy guy in Canada did the first studies in Parkinson's. But that was all NIH-funded research that got put together to make this treatment. And we don't do a good enough job of tracking, of saying, okay, this may be a great treatment, but let me tell you where it came from. It didn't come from Roche all by itself. It didn't come from Medtronic all by itself. It came because of discoveries that were made with NIH dollars, which were then used by those companies to make something that's good for patients.
Yeah. Oh, I just want to say another thing we do terribly. So we're all talking about how great we do. I actually think we do a horrible job in one of the most important things, and that we do need the public, and an educated consumer is who we want to be dealing with. And I don't think NIH has done a good job in educating from K through 12 in science education. I don't care if it's permitted or not, then let's go fight. Because the bottom line is that's where the nation's future is. That's where biomedical research's future is, not only in the support of what we're doing here, but where the kids are going to come who are going to take our places in this room and on this campus. And I really think we've not done a good job there, irrespective of how many times has story been told you can't do something and went off and did it. But anyways, the bottom line is I really think that is an area. Not being a director, I can say things like this. That is an area where I don't think we've done a good job, and I think there's a huge gap and a huge danger just watching the trends in the United States and around the world of what's happening in science education. And ultimately, that could be the downfall of this enterprise well before some decision in Congress.
Yeah. So I agree with all that. But the point I would add is that the responsibility for informing the public, which I agree, a well-informed, was it Sims who used to say, a well-informed consumer is our best customer or something. The responsibility for informing people about the exciting advances is all of you. It's every scientist. And I had a pretty dramatic example of that in an episode that occurred in the clinical center this past year and a half, where we had this epidemic that you probably read about of Klebsiella, which was carbapenem resistant, so the bug is resistant to all antibiotics. And 19 patients were infected, and 7 of them died. And then there were 10 patients who became carriers. So this was horrible. It was horrible for the whole community. But in that setting, the intramural program really created an opportunity, and it was led by a couple of PhDs in the Genome Institute, Snitkin and Julie Segre, and a young hospital epidemiologist in the clinical center, Tara Palmar, as the major leads, who said, gee, maybe we should do whole genome sequencing on these bugs to track this epidemic. And it turned out that this was a novel and new approach for hospital epidemiology and for tracking organisms, not just within a hospital, but between hospitals, between cities, and between countries. It's probably going to revolutionize the approach to this component of infectious diseases. But then they had to get up in front of the press and talk. Now, Julie had never spoken to the press, nor had Julie. But they did it, and they got out and they spoke, and it turns out people was sensitive to their way they spoke, and they did a fabulous job, and they felt good about it, and soon what was lemonade, or was lemons, became lemonade. And I think people realized that although it was a horrible story, people died, it also resulted in something useful and that was a contribution. So you got to go out and speak what you do, and you have to be willing to do that. That's part of your responsibility. It's all of our responsibilities. And I agree, you got to go to the elementary schools and do it, which I'm doing next week.
All right. Well, listen, thank you all very, very much for a very exciting, enthusiastic, informative, and optimistic outlook. Thanks again.